and we'll just let people drop in as they come along. I want to welcome you to the Centers of Excellence, Meet the Expert, Centers of Excellence in Molecular Hematology, Meet the Experts seminar series. Today our speaker is Jun Liu, who will speak to us in a minute, and I will introduce him. I'm going to just set a couple of ground rules for the seminar so that everybody is aware. We're going to ask people, you can show your video, but we will ask you to be muted during the presentation, if you would, please. And if you don't, I'll probably pop in and mute you. So thank you. The second piece is I'm going to take a minute to just take you to our website. So I'm going to share my screen for a minute. Let me get there. Okay. Can you see my screen? Okay, perfect. This is the website for the coordinating center for the Centers of Excellence in Hematology, U54 mechanism. And this is our group website. If you go, it's very simple, CCEH.io. If you go to this website, you'll find lots of informative information about the centers. The centers are listed here, which you can see. You can also find services that are available at those different centers by clicking on this service link. It's kind of being slow to get there. I'm not sure why today. There we go. Okay, and this will demonstrate, this has some tabs where you can go through and see all the different cores that are available to investigators across the country for doing hematology research. For example, if you click on, let's see, a genomics core right here, you'll find that the Cincinnati Center has a genome editing core, a DNA sequencing genotyping core, and an experimental hematology cancer biology core. So I hope you'll go visit this website. The homepage tells you about our seminar series, the Meet the Expert seminar series here, but also we have a monthly Microscope Monday seminar series. And the virtue of the monthly Microscope seminar series is if you have an interest in doing microscopy, and there are 3 centers that offer microscopy services through the U54 CCEHs, you can come to these seminar series and you can let your students, anybody can attend, and you actually learn really about the details of the kind of microscopy that you might want to use, get some advice. And also if you're, a person who doesn't understand the differences in some of the microscopy, this seminar series is a really educational opportunity to learn about what microscopy you might want to do in your research and the cores or core that you might want to use. I'm going to stop sharing. Also on this site, one more thing, there are funding opportunities. Let me just link to there. And what you can find here is Type A and Type B P&F awards. The Type B awards are closed right now, but we just had an application cycle that ended August 1st, and those are under review right now. But Type A grants are available to apply for anytime, and what they really are is core credits. So if you can write a proposal that's a one-page proposal about a core that you want to utilize, you can apply for what we call free money, essentially up to $12,000 worth of core credits to use a core at any one of the centers that would help you, get a preliminary data for future applications for extramural funding. I'm going to stop sharing right now, and then I want to introduce June to everybody. Let me get my details here. So June comes from us. Dr. Liu comes from Yale School of Medicine. He got his bachelor's degree at Nanjing University in biochemistry, and then went on to Boston University, where he received his Ph.D. in biochemistry. He then went on to a postdoctoral fellowship in Todd Golub's lab at the Broad Institute, and has been a faculty member at Yale University School. Yale University School of Medicine since 2009, and is currently an associate professor in the Department of Genetics. And today his talk is going to be on functional screens in hematopoiesis. I'm going to just reiterate to you, please mute yourselves. You're welcome to be present video-wise, but if you will mute so that we don't have feedback during the presentation, that will be great. Dr. Liu has agreed to answer questions during the seminar series, so please feel free to interrupt. If you're uncomfortable with that, you can populate a question in the chat. I am happy to have you read the question or invite you to read the question or read it myself. You just, whatever is comfortable for you. And thank you again, Dr. Liu, for presenting today.
Thank you, Diane, for the very kind introduction. And as Diane said, that if you have any questions, feel free to pop in at any given moment. I'm very used to being interrupted in the middle of talk and answer questions. I think that's more interactive overall. And the second thing is I probably wouldn't be able to see the questions in the chats very easily. So Diane, please help me to remind me if there are questions there.
I will do that.
So, thank you. So today it's a great pleasure to tell you some of our own experiences and also our own lessons about functional screens in hematopoiesis. And the reason, it's a very interesting topic for me to present here. There are several reasons. One of the reasons, of course, we have done ourselves quite a number of functional screens, try to identify genetic controls in hematopoiesis. The second reason is that the functional screens are becoming much more popular nowadays compared to used to be like 10 or 15 years ago. And the third reason, of course, is probably the most important reason here is that the Yale version of CCEH, the Yale Cooperative Center of Excellence in Hematology, or what we dearly called YCCEH, is offering a number of important services related to genetic screens that can be performed in supporting community work on hematology, and particularly benign hematology if you have any interest. So we have, for example, genetic library, viral library service where we can provide CRISPR libraries, for example, in both plasmid and the virus format. We have offer consulting on CRISPR based technologies, and we also provide the service to produce high titer virus for the community as well. And on top of that, we also provide the free consultation if you have any of your projects that you need help or getting a second opinion about designing your project. Please feel free to reach out to me, and I'm very happy to do this with you. And it's completely free, so hopefully some of you may find it useful. Now, what I'm planning to tell you today is really thinking from the genetic screen point of view, are sort of combining our own research experience ourselves and showing you several stories on that front, and really also what's the lessons we have learned through these phases. And one thing very important to say is that there are plenty of excellent work on both hematopoiesis and on genetic screens, as well as in the specific areas we're going to talk about, for example, on microRNAs and on genes such as TET2. I know there are several experts here, and I may not have a chance to mention all your work, but I do want to acknowledge at this moment that there are plenty of those work in the literature and in our field, and those are beautiful work, and which I may not have time to mention all of them. So if you think about genetic screens that can be done in hematopoiesis, and actually in general, overall in mammalian cells, there are two major formats people have been using to do genetic screens. The first one is more easy to understand, which I put on the bottom, which is so-called single well-based assays, right? So basically you have a multi-well plate, such as a 384 well plate. You have some cells in each of the wells, and you put one well, you put one genetic perturbations into that well, then look for what's the outcome of those individual perturbations. But this can be done in high throughput format so that you can actually assay many different genetic perturbations at the same time. Much more popular nowadays is the pool screen. So the single well-based screen used to be the most popular one. I would say about 10 years ago, this is probably more popular than the pool screen, but nowadays the pool screen is much more popular, relatively speaking, compared to single well-based screens. And the point of pool screens is, in essence, you're using each individual cell as if it's a single test tube or as if it's a single microwell to look for the consequence of genetic perturbations. So I'll first mention about some of our own efforts in this area and our own lessons in this area. Just to, as a start, this is not something that we invented in any way. This is actually the modern version of doing pooled screen really started from 2004 when René Bernard's lab started to use a pooled SHRI library to find the genetic controls in mammalian cells. In this particular study, they're trying to identify genes that control the p53 mediated network and in controlling of cell behaviors, in this case, cell cycle arrest. And they basically made a library full of many different shRNAs using a now very famous vector, which was still one of the most famous vectors used in shRNA called p-super. It's a retrovirus vector that can be used to infect cells. And these viruses will integrate into the genome of cells, and then they can use the abundance of the number of copies of these viruses or these shRNAs in the genomes as a surrogate for the number of cells that reflect the changes of phenotypes of the screen. And then they can read out the identity of the shRNAs using microarray at that time. So they designed microarrays, custom microarrays to read out these shRNA RNA sequences and use that to deduce very quickly which gene perturbation can actually cause changes in the phenotype of the cells. So my lab's own sort of exercise into this area started with our interest in regulation of hematopoietic progenitor cell expansion. Right, many of you know that, well, I think most of people here are the experts in the hematopoietic system, but I noticed there are a few that are trainees, that are new trainees in our own institute. So just very quickly, you have the hematopoietic stem cells, which are largely dormant in the mouse or human, that give rise to the mature blood cells. In the process, the progenitor cells are really the heavy loaders in terms of carrying out the majority functions of expansion of the system. So it's very important to understand what are the genetic controls of this progenitor cell expansion process. So at the time we decided, say, okay, we're very interested in microRNAs. I know a number of you here are also expert in microRNAs, such as Lee. At that time, we were asking a question, are there microRNAs that can regulate cytokine sensitivity in the hematopoietic progenitor cells? Why it's important? Because hematopoietic progenitor cells are powered in many ways by cytokines, and the cytokines are signals that allow the cells to proliferate. And those concentration of cytokines can basically, sensitivity to them can control basically how well the cells can expand. And to answer this question, we used a fairly famous cell line called BAF3 cells. And this BAF3 cells has been previously used to screen for cytokine independence by very famous oncogenes, such as BCR-ABL and the JAK2 mutations. And the BAF3 by itself is a murine, it's a mouse cell line. In the literature, many people refer to as pre-B cells, although I completely disagree with that characterization. I always think that BAF3 cells resemble mostly myeloid progenitor cells. They're dependent on this very important cytokine called interleukin-3 or IL-3. And with a good concentration of IL-3, they're happy, live, proliferate, and doing everything very nicely. And they're proliferating super fast. And when I mean super fast, they, for us, we have to split 1 to 50 for cells for every 3 days so that they can proliferate. So they really double very, very quickly within every single doubling is less than 10 hours. So it's a very, very quick proliferating cell line. If you don't give them IL-3, the cells are all dead within a few days, within 2 or 3 days. And if you give them a low concentration of IL-3, they are very unhappy and mostly dying, but they're unhappy. But this is actually a condition that allowed us to do our genetic screens. So basically, we're asking question: if we overexpress any given microRNA, can we find some that can actually induce the BAF3 cells to become happy under this low IL-3 culture condition? As a way to answer your question. Yes.
This is me in my naivety. Who's making the IL-3?
So IL-3 is that's actually a very good question. I don't fully know the answer myself. Maybe someone here in the audience actually knows. I only know that IL-3 is very abundant in the bone marrow environment, but which cell types, specific cell types makes them, I actually don't know. I know there are some stromal cells that can produce IL-3 in culture. That I know, but I don't know which cell type in vivo is the one that actually makes IL-3 in vivo.
Okay, I can go look it up.
Yeah, sorry.
No, no problem.
Yeah. So the way we designed the screen is saying that we take the BAF3 cells and then we'll put microRNA overexpression library and we'll make it into the virus format. The benefit of that is now we can control the titer of virus so that each cell will contain only one genetic perturbation, in this case microRNA. And they are labeled with different colors just to symbolize different identity of different microRNAs in here. And then we'll subject these pool of cells to a low IL-3 selection condition. So most cells will be unhappy, many cells will die, but there will be some cells that survive. For example, if the green microRNA or purple microRNA, blue microRNA, they're not doing anything to the cell, so there will be a low level of them will be remaining at the end of the screen. So you can see that there will be the same number of cells afterwards. But if the red microRNA is making the cells much happier under this low IL-3 selection condition, you will see more numbers of these cells carrying this microRNA after the selection process. Whereas for the orange microRNA, which will be depleted in the process, if it makes the cells less happy under this condition, they'll be depleted in the process. And this can be read out if we have a way to read out the content of these cells in terms of which microRNA they have in the genomic DNA, then we can do so-called barcode readouts for their relative ratios between different microRNAs compared before and after screen. Then we can deduce which are the red ones that make cells happier or which ones are orange ones that make cells unhappy in this whole process. So that's basically the whole principle of the pooled genetic screens. And one of the real benefits is you can make everything in pools. You can make a plasmids in pools. You can just make one big plasmid prep. You can make a virus in pools. You can make a big virus prep in a single pool without having to do so many different viruses, for example. Now, to make this screen work, apparently we need a number of things to make this work in the first place. First thing, we need to make an expression library for the microRNAs. At that time, no one has done that, so we were first to make this genetic library. And we basically did hand-based cloning. We ordered lots of oligos to PCR different genomic regions containing microRNAs, and each of them were then combined, and we mix them together into a big ligation reaction. In this case, using a gateway ligation reaction, recombination reaction. Then we screen many colonies to identify those ones that are corresponding to the microRNA that we designed, and then we pull them together afterwards. So this is how we made the library at that time. Of course, nowadays there are slightly simpler ways to make that, and I'll mention later. So this is one important component to have this screening work is to have this genetic library. Second thing you need to do is to detect the so-called barcode abundance of the different genetic constructs inside the cells. In this case, we have used a microarray-like detection. So we have a different color beads as if there are different locations on a microarray and a tether to a detection probe, whereas the genomic inserts from the retrovirus will be amplified with PCR and they'll be hybridized to these beads. Then we can read out how much abundance before and after screen for each of the microRNA inserts that we put into the cells. So this is a second technical necessity to allow this to happen. And third, then after that, so we are able to do the screen. So this is actually the raw data of the screen that we did 8 biological replicates. So that's why you're seeing eight replicates here. And for each of 8 replicates, we run analysis to see what are the most consistently regulated microRNAs. So these are only the ones that pass the threshold of filtering by statistics. And you can clearly see there are a number of interesting candidates. In this case, one of them is BR125B. This is BR215, so on and so forth. But there's also one thing I want to point out to you here is that even for one of the highest hits, in this case, BR125B, you can see that different replicates are actually quite variable in the amount of enrichment you have after low IL-3 selection process. So you can look at the colors. The most extreme ones can close to be 8-fold enriched. So this is log 2 enrichment, so 8-fold enriched, versus these colors will be slightly close to 2-fold or less than 2-fold enriched. So there could be lots of variations in this kind of screens, even can for a relatively small library. This library we have is about 500 entities in the library. So that's a very important thing to keep in mind when you design your own screens. And of course, 125B is a very interesting microRNA. The reason is that this microRNA and with its family member, BR125A, turned out to be highly expressed in hematopoietic stem cells. So this is one of the experiments we did at some point before that we isolated long-term hematopoietic stem cells or short-term stem cells, multipotent progenitor cells, so on and so forth, and then look at which are the microRNA's patterns enriched in the stem cells. And among the stem cell enriched microRNA here, where my cursor is pointing to, that contains both miR125A and miR125B. I didn't have the 125B label, but it's somewhere in here as well. So both of these microRNAs belong to the same family. They are both highly enriched in hematopoietic stem cells. So that's why we immediately went after these microRNAs. And the first thing to test is whether we can confirm and that there are true hits in the screen. So we have the vector with the microRNA expressed, but also have the GFP. Then we can mix the GFP positive cells with control unperturbed, so-called Y-type cells. And then we can basically put them into the same culture dish and put them under low or control IL-3 conditions, and then read out their competitiveness, relative competitiveness through a GFP positive cell to GFP negative cell ratio. So it's a very simple assay that we can read out. And you can see here that both 125B or 125A can really lead to high enrichment during the culture under low IL-3 conditions, whereas the control vector don't do the same thing. All right, so these are confirming that's truly in the BAF3 cell line, these microRNAs are confirmed to have these activities through the screen. Then we can also ask a question, what happens with the primary bone marrow cells? Because BAF3 is a cell line and the bone marrow hopefully will be the same. And turns out they're the same, that we can do colony formation assay using regular sort of a spiking different concentrations of IL-3 or GM-CSF. And you can see that the cells expressing 125A compared to controls are not only having ability to give rise to colonies, even with very much lower concentration of the cytokines, but for the same concentration of cytokines, they also give rise to bigger colonies in general. So this is confirming that 125A and 125B can induce cytokine sensitivity in bone marrow, in primary bone marrow cells as well. And when putting into in vivo, we can test this in the mouse model directly, and you can see that there's a dramatic expansion of GFP positive cells, which labels the 125A expressing cells, whereas the control GFP positive cells in the control vector doesn't seem to show this expansion. So overall, this 125A and B induces myeloproliferation in vivo, which is one of the microRNAs that have this capacity to do so. So this is basically our very first foray into genetic screens ourselves. And there are a number of things that we have learned through this simple exercise of one project. And I sort of summarize here into these 1, 2, 3, 4, 5, 6, 7, 8 different categories that whenever you design functional screens, at least for in vitro system, that you have to keep in mind. And the first thing is cellular system. And in this case, we are using a cell line, which is very easy to work with. If you have primary cells, of course, will be a little bit harder to work with in terms of both of the infection rate and also how do you culture them for a certain period of time. Like a hematopoietic stem cells, you just cannot culture for a very long time. Another very important thing, which I sort of separated out, I didn't mention earlier, is really cell coverage. This is probably one of the most important thing to whether your screen will be successful or not. And the concept is very, very simple, right? So for example, if your library has 500 things, you have to have at least 500 cells to be able to screen one genetic thing per cell. But in reality, you need way more than that. There are two reasons for that. One reason is that when you have a genetic library prepared in a pool, the representation of each of the genetic components is not at 1:1:1 ratio, but some of them will be higher, some will be lower. So if you only have the same number of cells per construct, you are not going to be able to assay all of them. Number two is that different cells will have different viruses will have different integration sites in the genome, and they may have disrupting their local genetic context, or they'll be regulated differently because of genetic context. So you have to have enough representation in terms of cell numbers for the same genetic construct inside the library before you can reach any useful conclusions. Then the type of readout we have is basically changing biological changes cell number, which turns out to be the major readout that many people use as a pooled screen. The readout, most of them are based on cell number readouts. So this can be proliferation, apoptosis, migration, etc. I will mention a few more later on. Now the Barker library, which I'll mention quickly, but you can purchase pre-made ones, but you can also make your own, and I'll tell you how to make your own after that. And Barker detection, at that time we're using microarray type of detection, but no longer popular, so it's all by next generation sequencing now. It's actually much cheaper. At the Yale, our core is $3 per million read, paired reads, so it's much cheaper than even for qPCR. Nowadays many times we just do deep sequencing rather than do qPCR assays. And infection rates is very, very important. That you want to keep roughly 20 to 30% to reduce multiple infection per cell. I will show a figure very soon on this front. Although I would say in some cases, when especially your cell number is limited, so you cannot get so many cells, you can aim for higher, to say 50%, that's still doable. And we have done those screens as well. On the technical side, it's very important to reduce the amplification bias, that both in terms of the, when you grow up the plasmid pool, make sure you have enough bacteria to be transformed with those constructs, so you don't have only 400 bacteria transformed for 500 constructs, for example. You should have a sufficient genomic DNA as a template when you try to amplify for analysis. So you're not only analyzing 400 cells for 500 different stuff, for example. Cycle PCR is also important to make sure everything's in linearity. And then lastly will be replicates. So we need at least 2 biological replicates to make sure there's no false positives. But in most cases, we run way more than 2, many times 3 or more. In the past case I showed you is 8 different genetic biological replicates in that sense. Now, so these are the sort of the lessons we learned from this very initial in vitro screen. And then we became very interested to say, okay, can we, before I mentioned about our in vivo screens, there are 2 things I want to quickly mention. One is how to obtain your barcode libraries. So I'm very proud to tell you that the CCEH do provide these things for you as a service, that we can provide the human genome-wide CRISPR libraries and the mouse genome-wide CRISPR libraries. We're also in the process of making the mouse and human genome-wide CRISPR-A and the CRISPR-I libraries available for you to purchase if you need. And then we have a library plasmid pools, or we can give you library plasmid pools or the virus pools, whichever format you think will be useful. You can also get the libraries from Allergen. The Allergen has many other libraries you can order, so that's another source for that. Although for these things, we offer at a much cheaper price than Allergen, so you don't have to spend several hundred, like $500 or $600 for the libraries yourself. Now, many times we also want to make our own. And why do you want to make our own? Because all these existing libraries are pretty big in size. Most of them are genome-wide libraries, and some of them are more dedicated smaller libraries, but even those smaller libraries may not be what you want to look for certain genes. So it's important sometimes to make your own libraries. And actually, personally, I find it, most of our screens are done by personal library, even for protein coding genes, that we also do sort of custom libraries ourselves, because a smaller library makes lots of things much easier to occur. Whereas a genome library, although it's really fancy and nice, but technically speaking, it's much harder to handle even for cell lines. So many times we do make these libraries ourselves. And there are several different ways you can make these libraries. One way to make these libraries to order oligos, and you can PCR, or you can make a guide RNA oligo library of oligos, and you can clone them as a pool of individual oligos. So this can be done by hand. For example, you have 200 or 300 genes, this is completely doable by a single person within 1 or 2 months. You can also order oligo pools that are much bigger. And currently, I've used services that can offer up to 5 to 6 times 10 to the 5th different oligos in the same pool, and up to 300 nucleotides in length. You can order those oligo pools. And what you do after that is you can PCR the pool with a adapter sequence on the pool, and then you can clone based on those adapters into the vector of your choice. And then you can actually make a pool, the single big transformation, a big ligation, a big transformation, and to get everything done in the pools. And we have made our own pools, and it works pretty well. And we not to advertise for this particular company, but we have been the company we has the most experience ourselves is with Twist Bioscience. And I know there are quite a number of other companies offer similar things. We just haven't had our own experience, so I don't have any comments on those ones. There are also other strategies I'll mention quickly that we can use so-called molecular chipper in our laboratory, for example, to make these libraries if we want to look at certain aspects of hematopoiesis. The second thing I want to very quickly mention is really the relationship between infection rate and the genomic integration. So the idea is if you, for example, if you infect a cell with a GFP encoding vector and you see 50% GFP positive rate, it doesn't mean that all the cells only have one copy of the virus in the cell. So if all the cells have roughly equal infectability by the virus, which is an assumption here, you can use Poisson distribution to model how many, what's the distribution of copy number in the cell population. For example, if you have a 50% infection rate, which is 0.5 of the fraction of infection rate, what you're seeing is about 60-some% will have a single copy of the virus integrated, whereas 20-some% will have 2 copies and maybe 5% have 3 copies, so on and so forth. So the reason 20 to 30% is normally recommended is because this is a range that you have most single copy. If you have lower than that, you have to start with way more cells, which is may not be a good trade-off in that sense. Now, with our initial experiencing in vitro screen, then we decided that maybe we can also do this in vivo as well, which of course will be more potentially interesting because you're addressing a primary cell behavior inside the in vivo environment. And at that time, we were also interested in the regulation of hematopoietic regeneration after the system had been injured. And one of the major type of injuries that are often seen for patients is really injury to the hematopoietic progenitor cells due to chemotherapy drugs. For example, 5-fluorouracil, which is a chemotherapy drug heavily used for gastrointestinal cancers. And the 5-FU is very capable to wipe out or at least reduce dramatically the hematopoietic progenitor cell pool within a day. So this becomes an interesting question for us is if we injure the hematopoietic system with 5-FU, and which microRNAs may regulate the recovery of the system into the mature cells faster or slower. So that's a question that we decided to address. And to answer this question, initially we're thinking about a more conventional approach. We'll say, okay, let's take the bone marrow. We actually take the mice and we inject the mice with 5-FU, or then we take mice that are not injected with 5-FU, injected with the vehicle controls. And then we say we isolate bone marrow and look for microRNAs to see which microRNA got changed or not. And turns out that almost every single microRNA got changed in this experiment, which doesn't yield anything useful for us, which is not surprising because when you do 5-FU treatment, the cell number perturbed, lots of cells are perturbed, cell lineage got perturbed by itself, and also there are lots of cytokine got induced. So there are lots of genes got regulated just because of these secondary effects. So it's not complete surprise that we cannot pinpoint any major candidates just simply by looking at this. So then we say, okay, maybe we should do a genetic screen to find out which one it is. So this actually turned out to be something that is very new at the time. So at the time when we're starting the experiment, no one has ever published on genetic screens in vivo. At the time we published in 2012, there was just one paper just published slightly before us in a slightly different system on genetic screening in vivo as well for hematopoiesis system. So at the time, we have no clue how exactly to do this. One of the questions we have in mind is how many genes we can actually assay or how many genetic perturbations we can actually assay in the mouse in vivo in this case. So the experiment design looks like this. We'll take the bone marrow cells from donor mice and we'll infecting a library of microRNA expression library. Then after the infection of the microRNA expression library, we'll transplant them into different recipient mice that are lethally irradiated so that the stem cell can be engrafted. And then we engrafted the system for up to 11 weeks after the transplantation, during which time we sample the blood periodically for the blood cells, which we can then analyze their barcode abundance during the process. Then we treat the mice with 5-fluorouracil, and then we collected 12 or 24 days after 5-FU injury to see how well these microRNAs may control the rebound or the repopulation of the mature cells inside the blood cell system. And we also collect
2 questions in the chat. The
Sure, yeah.
first one is from Jean Liu. Do you want to go ahead and unmute yourself and ask the question?
How to control infection rate? Yeah, so the way to control infection rate is if you don't know very well what's the virus titer, the best thing to do actually is to use your self-interest, if possible, to just use different concentrations of virus, to different amounts of virus to test out, what gives you the optimal range. And because different cells have different infectability, so many times, the companies or other places will tell you the MOI. But MOI is so tightly linked to the cell type you use. Different cells, MOI will be, for same virus, the different MOI will be different. So that's something that you should test out in your own hands. That's the best. The second question is this infecting a certain subpopulation of bone marrow cells for transplant or total bone marrow? I think it's probably talking about this in vivo project. So at that time, we just used stem cell, progenitor enriched bone marrow. So what we did is we treated the mice with conditioned mice with FU, and then we collected the bone marrow cells, and then we use a FICO to purify it. So the cells that got infected are the remaining still proliferating progenitor cells that enriched. And the reason we didn't do a more cleaner way is because we can actually wait for 11 weeks to allow many of those short-term progenitors to sort of die out during to weaken their contribution during the process. By the 11 weeks, most of them will be from long-term stem cells or from short-term stem cells, some of the short-term stem cells. So we know that this will be heavily stem cell enriched at this point in this case.
Thank you.
Thank you. So with this, now we can, there's another question, is this, oh, okay. So with this condition, now we can basically go on and do the experiment. And what we did, so one of the first thing we really want to know is, we actually have no clue how many microns we should put in there. We know that we cannot put too many because most likely we put too many, there's not going to be anything useful. And the system would not allow us to screen for many microns at the same time. So what we did is we picked 135 microns, and these 135 microns are highly abundantly expressed in different populations of hematopoietic cells that we previously analyzed their expression. So we don't know which one works, so we just combine these 137 together, 35 together, and put it as a pool to do this, without even knowing whether 135 is a good magical number to start or it's a really bad number to start. But nevertheless, we did the experiment to see what happens. So really the first thing to understand is really how is this particular system really basically putting the genetic perturbation into the stem cell populations, mostly in stem cell enriched populations, and how many genetic perturbations we can actually assay at the same time in a given mouse. So this is sort of a plot to try to answer that particular question. So here we did three different cohorts based on 3 independent infections. Each independent infection, we go into 5 individual recipient mice. And then we are assaying the peripheral blood from week 3 to, in this case, week 11. So different bars are for different weeks. And the red one is for post-5FU. So that's what happens after 5FU. So you can see that there's a dramatic reduction initially in the number of microns we can still detect, or cells that we can still detect within that are integrated into the genetic system. And after a few weeks, that starts to stabilize. So this is actually more consistent now with some more understanding recently from additional sort of genetic tracing experiments, that there's a certain limitation by which how many cells we can genetically label in the hematopoiesis system in the mouse during the transplantation process. And in our own hands at that time, due to also limitation, we don't want to get 100% infection rate, right? So it's probably somewhere around 20 to 30% infection rate. We can roughly assay, if you want to read out per mouse, you can read out at the end roughly 40 different barcodes or different perturbations per mouse you can read out in the bone marrow system. And we can also look at how many different mice will contain, how many barcodes are shared between all the mice or some of the mice. So for mice that contain all the mice, we have total 15 mice. For all the mice to have the micron in there, it's actually very few. It's less than 10 of the microns are present in all mice. Whereas if you look at anything that have 4, 5, or 6, you can have maybe 40 or 50 different microns. So now, of course, we know better, and I think this will be the number to shoot for, is if we do in the future similar things, we will probably limit ourselves to 30 or 40 perturbations per as a pool of library, and you can repeat different libraries in the mouse for those screening purposes. And another useful information for you to know is that if you simply look at barcode abundance without doing any computational analysis to compare, the really differences between different mouse. Different donor mice are different in their barcode presence and availability, which is consistent with the idea that when we use, for example, 130 something to go into the mouse bone marrow system and do a transplant, lots of these cells, there are only limited number of stem cells will be able to engraft. And those selection of engraftment will be random, so that they will randomly select among the 135 which one will present at the end. So that's why the barcodes for the mouse will be sort of selected based on that, based on randomness. So that's why you cluster the barcodes based on the mouse. Now, of course, we can still do something intelligent based on this data, even though they're clustered by the mice, is that we can compare the same cohort mice before and after 5U, or actually the same mouse before and after 5U, and compare the same mouse barcode increase or decrease. So if we compare that, this is sort of a figure that we plotted. So in gray are the microns that we didn't detect at all in those mice. So you can see actually there are lots of microns we cannot detect. Some of the microns we cannot detect in any of the mice. Some of them we can only detect in a very small number of mice. So those are not very, very useful. In the blue are the microns that are depleted during this process. And in the red are the ones enriched. And in the white are the ones we can detect, but didn't change significantly, either increase or decrease. We have a threshold cutoff. So you can see that data is much more noisier than the in vitro screen that we present. And part of the reason is, I think, because we started with 135 different things rather than maybe 30 or 40, which probably would be better. But nevertheless, we can still read out interesting things. And the most consistent thing is not on the right side, which are the enriched ones, because those enriched ones you only see sometimes in one cohort, but not in the other cohorts. Whereas the depleted ones, commonly depleted ones, seem to be shared among many different cohorts, so quite a number of different cohorts that we can actually read out the depletion of these barcodes. Meaning that when these microns are overexpressed, they are likely to induce a suppression of the hematopoietic recovery process. So we took several of them to validate, and we can validate. And one of them is this micron called miR50. And when we overexpress miR50 and look at the recovery of 5U compared to controls, the miR50 have a really reduced recovery rate in the peripheral blood. Expressing cells have a reduced recovery rate in the blood for myeloid, platelets, and to some degree B and T cells, although it's not very as significant as these myeloid platelets. Then we can ask a reverse question: if you knock out this micron, do you see opposite effects? And yes, we do see some opposite effects. So this is to knock out miR50, and we're comparing wild type versus knockout cohorts, and you can see that the knockout cohort do recover better for the myeloid and platelets. And we don't see any significance for B and T cells. So at least for the more myeloid side, yes?
It's a question from me in my naivety again. Are there certain microRNAs that you never see go in?
Yes.
Like if you go back 2 slides.
Yeah.
Right here, it looks like there's some sort of areas where it doesn't look like it is detected well, or
So I see your point, Diane. So I think you are trying to allude into a point is whether there are microns are against engraftment itself, right? So basically, if you overexpress microns, they'll be so against engraftment, so you never see them afterwards. We actually don't know the answer to that. Part of the reason is because at the time the screen was done with probably too many microns. So there are lots of random dropouts due to the effect I described that sort of make us hard to statistically conclude which micron may be doing this on their own. So I think in the future, if we redo this experiment, we would do a smaller pool, and in the smaller pool, we'll have much better chance to answer that question than using this bigger pool.
Great, thanks.
Yeah, so overall, so through this in vivo genetic screen, so the concept is, yes, in vivo genetic screens can be doable, but there are way more limitations compared to in vitro genetic screens. And when I say way more limitations, really it's system dependent, right? So if you're asking a different system, for example, if you're working with T cells and you're harvesting lots of spleen T cells and then inject into mouse and only assay for a short period of time, that's actually a very doable experiment, and many people have done T-cell-based screens. But at least in the hematopoietic stem cell pool, there's a major limitation on how many things you can sort of assay with confidence during in this kind of experiment. So there need to be sort of optimized coverage in this case, and you need to understand your system better to be able to do this. And cell coverage often will be limiting because you don't have as many cells to start with, and the same biological readout. And so the key thing here is, because of these noises, it's likely that you need more biological replicates to allow this to occur. So this is our sort of experience with in vitro and in vivo screens, both with sort of biological readout as a cell number in this case. One is the in vitro low IL-3, one is the in vivo recovery after bone marrow transplant, 5FU insult. There are other things we can assay as well. For example, you can assay for gene expression readout. So this is actually a beautiful paper done on BCR11A, so the regulatory region for BCR11A using CRISPR, that the authors that designed the CRISPR RNAs against a region that potentially controls the BCR11A expression. Just for those of you who haven't heard it before, I think most of you definitely know this. BCR11A is controlling the adult and fetal to adult hemoglobin switch process. And in sickle cell disease, if you increase the fetal globin, you actually can sort of reduce the symptoms in those patients. So they use this one to basically read out, which genetic regions in the genome are controlling the expression of BCR11A in this case, and also consequently fetal hemoglobin as well. And we sort of, at a similar time, we actually started our project before they published their paper, but we actually had a similar interest to use the genetic screens to understand regulatory regions for things. And we've been interested in this particular microRNA called the miR-142. And for a number of reasons, miR-142 is one of the very first microRNA ever cloned in the hematopoietic system. And the second thing, it's actually have a reverse pattern to the 125A and B. 125A and B are sort of high in the stem cell and gradually dwindling so down to the mature cells, whereas 142 is, I would say, quite specific, not complete, but quite specific to the hematopoietic system, but it's actually low in the stem cells, but increase during the differentiation process. So this is a very interesting microRNA, and there are knockouts from miR-142 now that really shows several important functions for miR-142 in the hematopoietic system. At that time, it wasn't clearly known, but nevertheless, we saw it's interesting microRNA. We try to see whether we can understand the sequence that can control the expression of miR-142. And the way we designed this, the effort is a very similar idea as a hemoglobin study, that we try to design many guide RNAs tiling the whole region that we think might be controlling the expression of miR-142. In the red here are the two strands from the hairpin of the microRNA hairpin for 142. So we designed these different guide RNAs on both sense and antisense strands. And then we can, so we envision a screen where we can put the cell population with high expression of 142, miR-142. Then we can put the different guide RNA into the system. And if those guide RNA hit a region very important for the regulation of 142, they will probably decrease the expression of miR-142, so that we can use a sensor. If there's a low miR-142 expression, the cell will turn green. Then in this case, then we can sort out cells that has a green signal, means they have low 142, and compared to high 142 expression cells, which are colorless, then you can compare to see what's the representation of different microRNA or the different guide RNAs in there, CRISPR guide RNA, and to understand which region of the 142 will be important in this process. So one way you can do this is to by complete design by hand. They can use a pool synthesis to do this. At the time we say, okay, are there other ways we can do this that's better? Wouldn't say better, but at least cheaper to do and easier to do in the lab. And we found a way that we term it called a molecular chipper. And the reason we name it chipper is because we got motivated by these machines that often appear in our neighborhoods because of the winds that bring down trees, that they take a piece of wood and they make it into smaller, even-sized pieces. We say, oh, that's something we can do with genomic DNA, right? So we envision, say, okay, we have these pieces of DNA we want to perturb as regulatory regions, and we want to produce very dense guide RNA from these regions. How do we do that? So what we decided to do is that we can go through a molecular process that's just like this machine, but a little bit black box for you now, but I'll explain very quickly what it means. Then we can, after we can produce these fragments randomly of the right size, then we can clone them into a guide RNA library and be able to use this as a perturbation to quantify the effect of miR42. So the black box turns out to be not that complicated. So we actually utilize enzymes, type 2S restriction enzymes, where they will recognize a sequence, in this case, EcoP15I sequence, and that will cut outside the recognition sequence for about 20 nucleotides that we can grab. So the idea is we'll take any piece of DNA we want to quantify or want to assay for. For example, it could be many different PCR fragments, could be a back clone. We have done all those. And you can randomly fragment. So the idea is we want to grab the random ends of those fragments to make them into a guide RNA library. And so we did that, and it's doable molecularly. It's actually not too complicated. And also very, I would say, much cheaper than you order oligo pools, and potentially faster too, because you have to wait like 10 days for the oligo pool to arrive, during which time we can probably get this done already. So with this, we use the BAF3 cells to do the screening. Again, BAF3 become our friends here because it has a high expression level of this microRNA code 142, and it also happen to have a very low expression of the other microRNA I just mentioned, 125 and B. So we designed a reporter to report the expression of the microRNA expression levels, so that they have after the GFP they have 4 binding sites for this particular microRNA 142. So if the microRNA is high, the GFP will be very low because they got inhibited by these microRNAs. But if the microRNA is low, GFP will be high. So these are our basic cells without the library. After we infected the library, what we get is positive cells coming out from here. So these are the cells with very low levels of miR42. Then we can sort these cells populations out, and then we can do sequencing based on that. And after doing that, we can come up with regions that are basically, if you have guide RNA in those regions, they can actually hit and regulate microRNA expression. These are places where the mature microRNA sequences are. So of course, no brainer, you should hit those ones, because if you inactivate those regions, the microRNA is not produced. But we're actually quite surprised you also find things here and here that seem to be consistently hit by different replicates. To make a long story short, what we found out is these regions actually control the microprocessing to make these microRNAs actually possible to be produced in the first place. Now, so from this exercise, what I'm basically telling you is that it's also clearly possible to use gene expression as a readout. In this case, we use the reporter for those microRNAs. But we are not using gene expression directly. We're converting the gene expression into cell numbers, enrichment of cell numbers, because using the fluorescent reporter to sort of cells. So at the end, we still assay for cell numbers, but we're using gene expression reporter to help us to do the cell number enrichment in this case. So there are now some new developments that you can use more elegant ways, so that potentially you don't need to develop a reporter for any gene that you want to screen. So this is a work by Stephen Riley, who recently joined our department. They use so-called hybridization chain reaction flow fish. So the idea is for any given gene expression target you're interested, for example, you're interested in a particular gene in the hematopoiesis system, you can design an assay so they can use the flow to quantify the RNA expression in individual cells. With that, you don't need to make a knock-in or make a reporter for that anymore. So you can just directly do that. And in theory, you can also potentially do more than one color. So you can measure more than one color or one gene at a time, potentially. So they claim that this can be done with very lowly expressed genes, such as those with transcripts per million at 1 transcript per million. They can also do that to some degree. So that potentially will be a very interesting way to move forward for gene expression-based readouts for cells, at least for a single gene readout in this case. There are also efforts that using single-cell RNA-seq-based readout for screens, and I actually wanted. So we are doing something similar to this right now, and I thought we'll have data for you today, but it turns out we're a little bit delayed, so we didn't have data for today. But I'll just quickly go through my understanding of the system and what we are doing, thinking which way we'll follow ourselves. So as I mentioned, as a screen readout, you can have cell number readouts, which is increase or decrease by apoptosis, proliferation, etc. You can also read out single gene expression by converting to cell number via cell sorting. But in this case, you're reading out the many genes expression in the single cell RNA-seq readouts. And this is one of those papers from, in this case, from Yale Image Lab, where they can simultaneously understand the gene's function in multiple cell population, in this case, monocytes and the dendritic cells, and try to assay in the same cellular system using single cell RNA-seq as a readout. And there are multiple ways, vectors. There's Regev lab, Regev's lab, Jonathan Weissman's lab, and the Image lab have their own vector systems. And there's another vector system called crop-seq vector, which is much more popular nowadays because it's easier to recover the guide RNAs and the fact that that they don't get scrambled. So one of the things to be able to read out the identity of guide RNAs, we need the guide RNA to have close to the poly A tail to be able to read it out. And in these studies, they all inserted a barcode that's away from the guide RNAs to read out the identity, which they previous have a dictionary so that they know which guide barcode is corresponding to which guide RNAs. But the problem with this approach is because they are quite far apart. And during a pooled virus preparation process, there are sometimes template switching. That the barcodes got hooked up with the wrong thing during the virus preparation process. So crop-seq vectors tend to reduce this effect dramatically. So that's become a much more popular vector for doing this kind of work, and that's something that we're using currently to do so. But with this, one thing that we've been thinking about really the issue of the scale and the cost of doing that. And it turns out that it's not as easy as you think, as fancy as you think could be, because you still need roughly 50 to 100 cells, and actually many people do more than 100 cells per guide RNA, CRISPR RNA. You need 3 or 4 CRISPR RNA, or sometimes more if you're doing CRISPR-A screens per gene. And you also need many control guide RNAs. And that coupled with the fact how many cells you can really read out from single cell RNA-seq reads, you're realistically, in many cases, if you read out, for example, 20,000 cells, which is quite a bit already, you can only assay the function of 15 to 30 genes maximum in those systems, even in an in vitro system. So that's a sort of a potential limitation. And the one thing that can potentially improve that is by using target enrichment, so-called TAP-seq. So you only sequence a subset of targets rather than all the genes in the genome through a single cell RNA-seq. And they claim it to be about 10-fold better. So you can read 10-fold less reads to be able to do the same genetic screens. And we're doing that right now. We don't have data yet, but hopefully in the future we have that. And I think I have probably have slide over time, but very quickly, I want to mention last point, which is functional screens in single cell well assays. It's no longer as popular, but I do want to emphasize there are some very important features that for some of your things you're looking at, maybe it's actually very helpful to do. And the major advantage, disadvantage, of course, you have to have a library that's already arrayed. You can, each individual well will contain the individual component of the library. But the advantage is really you can read things other than cell numbers and gene expression, right? For example, one of the things we're interested to do is cell morphology, and you can actually use this to read out cell morphology on individual wells. You can also use this to read out things where the functional effect is relatively small, and I will show you one example very quickly. That with the pool screens, your effect size will need to be a certain size. I think at least 3-fold or more to be able to reliably read out those functional regulating genes. But for this kind of assays, you can actually read out 30%, 20%, sometimes 10% differences, and it can still be reliable. And one of the things we did at that time was trying to find the TET2 regulating microRNAs. As many of you, TET2 is a very important gene, and it's dose dependent in the hematopoietic system. So at the time, we were very interested to find microRNAs that can control TET2 gene expression. And what we decided to do is to pair 3' UTR of human and mouse TET2 with individual microRNAs, assay them individual pairs in 384 well plates. It's easy for me to say, but in reality, we're using robots to help us to do this. Otherwise, it's very tedious to do. But the benefit of doing that is now we can actually read out small differences. For example, this is control where there shows no change of the 3' UTR activity. Now we can actually read out many different microRNAs that can only regulate target genes by target 3' UTRs by 20–30% effect, and then we can still get reliable data from that. So that's very different from post-screen. That post-screen is if you only have 20–30% effect size, forget it. It's not possible to do screen. But with the individual well assay, it's actually possible to do. And through that, we found many TET2 targeting microRNAs, and of course, we confirm many of them have functions. So I want to quickly finish here is saying that with single well assay still is a viable approach for certain use cases, especially those things that you cannot read out for cell numbers or by gene expression, or those ones you expect to have a low functional effect potentially. So that could be still very, very helpful in that sense. I want to stop here and really want to acknowledge CCEH that allow me to do these things and support me. And also we provide these services to the wider community. And happy to, if you have any questions or you are doing screen, you want to get our opinion, feel free to send me an email.
Terrific. Thank you so much for that. Anybody have questions they want to ask, feel free to show yourself and ask your question. Clear for everyone. Complicated for me. I'm going to own to it.
Yeah.
I'm thinking about the application of it.
Yeah, so as I mentioned earlier that we provide services, free consultation for that. So any one of you who are thinking about doing screens, feel free to give us a call or send us emails. We can arrange time for a call to discuss the details.
Awesome.
We're happy to share our experience. Yeah.
Okay. Anybody else want to ask a question? Hey, Amy, nice to see you. Okay. I would want to just remind you that if you want to review the talk that's been given today, it will be posted on the website in the next few days. Thank you all for your participation. And thank you again for your presentation, Dr. Liu. It was really good.
Thank you, Diane. Yeah.
Okay. Take care, everyone.
Thank you.